Thank you so much to the organizers, especially Connor and the EPIC seminar um, organizers and sponsors and SGP for this fantastic opportunity to showcase my postdoctoral work here in Henry Coldcraft's lab at Columbia University. So I'm really excited to share with you all an overview of what I've been up to with studying cardiac ion channel trafficking regulation today. So let me jump into the. Okay. So I'd like to start off with a small thought experiment to articulate the problem that I work on. Imagine getting dropped off at this beautiful yet crowded city and asked to go from A to B with no GPS or directions. I know in my own case, I'd likely struggle, despite this being my hometown in southern India. However, if I happen to have a set of directions, then the problem becomes tractable. So this is very similar to what cell surface localized membrane proteins like ion channels face. They are born somewhere in the ER. How do I do a pointer? Okay. So they're born somewhere in the ER, and then they have to navigate this really complex intracellular milieu to reach its functional location at the cell surface. And then at appropriate times, often in the order of hours, they need to be internalized back into the cell to be degraded or recycled as needed. Now, this has to occur in a very precise fashion, as dysregulation leads to devastating trafficking disorders like cardiac arrhythmias, neurodegenerative disorders. My research primarily focuses on elucidating the mechanisms and signals that prompt an ion channel where it needs to go within a cell, how dysregulation occurs in disease, and to be able to use this insight to develop therapeutic strategies to rescue trafficking and functionality in these conditions. So ion channels need no introduction to this group. They're really essential regulators of physiology, and very unsurprisingly, they're critical drug targets for various disorders. Now, some examples, these diseases are different, they affect different organs, manifest distinct clinical presentation, don't really have much in common at first glance, but they have shared mechanistic underpinnings, mutations in ion channels that result in decreased functional expression at the cell surface. Now, the simple big idea is that if we can correct this underlying problem and rescue function, then it could be a transformative approach to treat many different conditions. So majority of my postdoctoral work was focused on the cardiac potassium channel, KCNQ1. And then I'm sort of splitting my talk into three topics encompassing past projects as well as some insights on ongoing and future research directions. So the first one would be developing proximity approaches to mimic adrenergic regulation of the slow delayed inward rectifying potassium currents, IKS, in the heart. So, taking a step back, cardiovascular diseases collectively are the world's leading cause of death, given the crucial role of ion channels in maintaining heart rhythm and contractility. These are key targets in many of these conditions. The ion channels that decorate the cell membrane of cardiomyocytes function together to coordinate cardiac action potentials that allow the heart to contract and relax. The potassium channel, KCNQ1, conducts the slow potassium current that's important to reset ventricular action potentials. This channel is typically a multimer composed of four pore forming Q1 subunits and two, at least, of the accessible E1 subunits. Loss of function mutations of IKS slows down repolarization, resulting in prolonged QT intervals and increases the risk for TDPs. And this risk is particularly high during exercise when there is an increased calcium influx by protein kinase A activation or PKA activation. This needs to be balanced by a substantial increase in IKS to maintain physiological action potential duration. Now, protein kinase A is composed of a catalytic C alpha domain linked to a regulatory R2 alpha, which is activated by cyclic AMP and then recruited to the channel neighborhood by binding to the kinase binding protein ACAP9. Now, mutations that disrupt this interaction cause long QT and loss of adrenergic regulation for these patients. So, we sought to develop a synthetic approach to mimic PKA modulation of IKS, bypassing the need for ACAP9 to address this condition. So, to do this, we leveraged proximity approaches, taking inspiration from phosphorylation inducing chimeras, which are really just small molecules that are linked to one another, one that binds a kinase, the other that binds the target protein, inducing their proximity and enabling phosphorylation of the target protein. Now, this, of course, has several limitations, such as specificity of small molecules as well as limited protein targets. So, we sought to develop a nanobody based technology that can bypass these limitations. So, essentially, we delivered PKA subunit, the regulatory subunit R2 alpha, to the channel neighborhood by linking it to a YFP GFP binding nanobody and expressing it along with the channel and heterologous systems with the YFP on the E1 subunit or a YFP on the Q1 subunit. Now, does this tool work? So the nanobody by itself, when targeted to E1, is silent. It transfected in CHO cells. It had no effect on IKS currents, even in the presence of cyclic AMP, because these cells don't have ACAP9. So we didn't transfect them in, basically. However, when we link it to the regulatory R2 alpha of PKA subunit, then we see an acute increase in IKS amplitude only in the presence of cyclic AMP. So this tells us that the idea works. However, the catch is when we deliver this regulatory R2 alpha directly to Q1 subunit by expressing it with the C-terminal YFP tag and using the same YFP nanobody fusion, here we see that the tool basically decimates the IKS currents. Now, we swap the regulatory subunit to the C alpha itself, which is catalytically active, and we see the same effect. We confirm that it was due to the catalytic activity, because when we have a dead catalytic version, this has no effect. So, using proteomics, we designed that targeted C alpha with the nanobody actually phosphorylates a number of different residues that are not modified by freely expressed uncaged C alpha. So, to gain deeper insight on the loss of current phenotype with nano C alpha, I probed the surface expression of the channel using flow cytometry and by engineering an extracellular bungarotoxin binding site or BBS into the channel that can be labeled with a fluorophore when it's exposed and is at the cell membrane. So what we saw was nano C alpha has a left shift in surface expression consistent with a significant reduction in channel surface density. Confocal analysis with subcellular markers identified an increased co-localization with the early trafficking compartment Golgi. So, to briefly summarize this work, it provided insights into how we can take PKA subunits and be able to achieve targeted adrenergic control of a channel of interest. So, distinct subunits have the subunits have distinct impacts based on site of recruitment, based on our data, E1 versus Q1. Now, we saw that the tool can mimic acute adrenergic upregulation when we target R2 alpha to E1, but when we directly target it to Q1, this impacts trafficking and arrests the channels in Golgi. So quickly segueing into the second and the major project. So we leveraged the conceptual framework and techniques from the first project to then set out to examine ubiquitin code of regulation of ion channels, which became the major focus of my postdoctoral work. So As electrophysiologists, we tend to we engage with channels when they're at the cell membrane and tend to think of them as static residents there, but our data suggests a very different picture. At steady state, Q channels expressed with a C terminal YFP tag are actually highly distributed throughout the cell when transfected in hex cells. They co-localize with markers to the ER, Golgi, endolysosomal compartments, pointing to a really dynamic equilibrium between forward trafficking, recycling, and degradation to maintain what we measure at the cell surface. So the question then becomes, what are the signals that guide Q channels through these distinct intracellular compartments? So, looking to literature, it's very well established that Q channels are internalized upon ubiquitination, specifically by the ubiquitin E3 ligase, NET42. This is a very prominent E3 ligase that's thought to regulate a number of cell surface localized channels and receptors. So, this allowed us to hypothesize that ubiquitin might be a key player in at least one or more of these distinct steps along the channel's life cycle. So, a little bit about what ubiquitin is. It's a post translational modifier, a highly versatile signaling molecule, because it can not only monoubiquitinate distinct lysine residues on a substrate, but it can also then go on and ubiquitinate one or more of its own lysine residues, forming polyubiquitin linkages of distinct conformations and shapes that can each code for distinct cellular functions. Now, canonically, protein degradation is linked to lysine 48 or the K48 link chains. It is for soluble proteins, as well as K63 is the other major linkage type, which is thought to be involved with protein trafficking. Now, all the other linkage types exist as well. They're collectively called minor linkages, but their functions are even less clearly understood, especially in the context of ion channels, because of their lower abundance and also lack of technologies to really manipulate them in a substrate specific manner. So, the primary question we started out by asking was, is Q polyubiquitinated when expressed in hex cells? So Dr. Scott Canner, who's co-first author on this manuscript, as well as the other one, is a fantastic colleague who worked with me in many different projects. He pulled down the channels from hexyl lysates and then did ubiquitin proteomics with the core facility here at Columbia. So we saw that the channel was actually basally polyubiquitinated, primarily by K48 linked chains, followed by a small amount of, followed by K63 chains, 24%, and then a very small amount of the minor linkages collectively together. So now the question is, what is the role of these linkages on Q? So to test that, we took advantage of the fact that ubiquitination can be reversed by a class of enzymes called D-ubiquitinases or DUBs. A handful of these have intrinsic preference for particular ubiquitin linkage types, K63, O1 for K63, O4 for K48, Cezanne for K11, Trabit 2933, U21, which is linkage non-selective. So we made engineered D-ubiquitinases by fusing catalytic domains of these DUBs to a YFP nanobody, similar to what we did in the first project, express it along with Q with C-terminal YFP, so that it can bring the catalytic function close to the channel neighborhood and hopefully remove specific linkage chains from the channel and then see how that affects channel behavior. So first of all, do the tools work? A lot of the biochemistry for this project was done by Dr. Eno Kamar. Really, biochemistry was our go-to tool to see if ubiquitination was reduced, since mass spec especially was not very good at picking up the minor linkage types in terms of signal to noise ratios. So what we saw is that Biochemically, when we pull down KCNQ1 channels co-expressed with either the nanobody by itself or one of the different engineered deubiquitinases or the NDUBs. We see robust channel expression. So Q1, because it's a tetramer, runs as monomer, dimer, trimer, and tetramer. And then we strip this blot and then reprobe it with antibody towards ubiquitin. So we see that basally ubiquitin runs as a smear because of the polyubiquitin chains that are of different sizes. So we see this dark smear for nano condition, uh, indicating high ubiquitin levels. But all the tools were pretty good at removing ubiquitin, with the non-specific one in blue, U21, really wiping off all the ubiquitin from the channel. So the surprise was then which chains were actually important. So, to look at changes in channel expression using YFP as a readout, the major chain, using YFP as a readout on flow cytometry, we saw that the major chain types on Q1, both K48 and K63, removing them had absolutely no effect on total amount of channel. Um, Surprisingly, when we remove the minor chains, K11, 29, 33 in pink and orange, here we see huge increase in amount of channel in the cell, and we see that also with the non-selective end up. So the next question is, okay, so is having more channels then sufficient to see more of the cell surface? The answer is not always. So while removing minor linkages in pink and orange increased surface residence, which we measured by labeling the extracellular bungratoxin binding site with Alexa 647 fluorophore, we saw that for the non-selective one, even though we saw probably the highest amount of Q1 expression, we didn't really see any change in surface levels of the channel in this condition. And then even more surprising is K63, which is thought to play a crucial role in modulating endocytosis and surface trafficking. We saw no effect for this channel in turquoise. Removing K48 in green actually had a negative effect, suggesting that this linkage is actually probably important to maintain steady state expression levels of the channel in hex cells. So we went ahead and confirmed if what we saw on flow actually translates as a functional change using electrophysiology done by Dr. So here we were happy to see that indeed there was an increase in function for the minor chains in pink and orange, and a decrease for the the K48 removing one in green, and then no change for the other two. So to gain to gain deeper insights into the mechanism at play, we adapted an optical pulse chase method from the flow cytometry assay I showed before. So here we label the cells with bungratoxin, so saturating channels already at the cell surface, and then we put them back at 37 degrees to allow for cells' normal functions to resume. So presumably new channels with free bungratoxin binding sites would be inserted. And then we put them back on ice, and then we label them now with fluorescently tagged bungratoxin. So we can specifically label the new channels that got delivered. And we do that as a function of time and measure them on flow cytometry. Plotting them, we were really excited actually to see that the trends we saw for forward trafficking nicely explain the differences we saw at steady state. So removing minor chains, orange and pink, enhanced forward trafficking compared to control in black. And then the non-selective as well as K48 actually had relatively slow rate of forward trafficking. So that explained why, even though we had just a lot more channels in the non-specific condition, they're not really getting to the cell surface quite as well as they should. And then we confirmed some of these results using confocal to actually see if our predictions are right, with doing co-localization analysis with the forward trafficking compartments, ER and Golgi. We did see an increased co-localization for the K48 one, which reduces forward trafficking, as well as we see that for Golgi. And then we see a decrease in ER. So we kind of see a lot more at the cell surface for the pink and orange conditions that enhance forward trafficking. So that was all to do with forward trafficking. What about endocytosis? So we did the same assay now, but we do the opposite where we first label with BTX bungarotoxin linked to biotin to the channels that are at the cell surface at T0. And then we put them back at 37 to allow for endocytosis to occur. And then channels that remain at the cell surface will now be labeled by streptavidin 647. So new channels will not have bungarotoxin biotin because we would have washed them away. So those would not be picked up in our assay. So here the results suggest that really the prominent player was actually K63 removing O1 in turquoise. The others with maybe a small effect for K11, not really significant change for the others. And we did the subcellular co-localization assays, and we saw indeed a slight deficit with the endocytosis compartment lysosome for O1. So what all of this told us is indeed K63 plays a role in modulating endocytosis, similar to its established function in literature. It's just that we didn't really see this to be enough to make an impact at the steady state level. We saw that that was because of some complicated way by which cell handles recycling, which I won't be going into today in interest of time. But you're welcome to see that in the paper. So that was all wild type channels. We then looked at disease relevance of polyubiquitin signals, whether this is important. So as I mentioned before, impact surface trafficking of Q1 is also a prominent mechanism that underlies long QT1. So we chose a handful of trafficking deficit mutations, all localized to the KCNQ1 C-terminal domain, and examined their ubiquitin responsive signatures by adding the linkage-specific end-ups to this mix. So for a subset of mutations, what we observed is that the minor linkage in orange, which removes K2933, was really the best at rescuing trafficking to varying degrees. For some other mutations, however, we saw that while it had a modest effect, the major player was actually K63, suggesting that even amongst mutations that cause the same disease, there could be really critical differences in the underlying mechanisms and what ubiquitin signatures may underlie it, informing targeted therapy. So this also points to the fact that a trafficking impairment could potentially also be because of reduced forward trafficking compared to enhanced endocytosis, hinting at mechanisms that we can target for particular mutations as a therapeutic approach. So in summary, the results of this study are consistent with this model where minor linkage chains, K112933, are operating primarily in forward trafficking, while the major linkage K63 is involved in endocytosis. Intriguingly, K48 in green was required for normal forward trafficking of the channel. And further, certain mutations likely have which have a forward trafficking defect are best removed by removing K112933, the minor chains, versus some other mutations which likely are just rapidly endocytosed are best rescued by removing K63 chains. So with these insights from hex cells, we wanted to study ubiquitin code of Q1 channels within its native environment to see if that makes any difference. So we isolated guinea pig myocytes and adenovirally expressed our channel with the suitable tags similar to the hex cell experiment so we can measure surface and YFP levels using confocal. So here, the results showed a completely different story, where we saw that total expression was actually primarily regulated by removing K63. So was the surface level, where we see enhanced YFP and K4 and 647 signals. The effects of removing K48 as well as K11 were similar to what we saw in Hexel experiments. However, the orange one, removing K2933, had absolutely no effect in this context on total or on surface. So these results suggest that depending on the cellular context and potentially maybe the type of E3 ligase is present that can have different polyubiquitin specificities, channels are regulated by distinct ubiquitin chains, and our tools essentially serve as a way to gain insight into this and evaluate what chains are present. Okay. So, however, the discrepancy between Hexel and the myocytes was really intriguing. So we knew that NET42 regulates KCNQ1 channels. So we went back to the Hexels to see if adding a different E3 ligase changes polyubiquitin signatures on Q1. So we chose NET42, which is a known modulator of KCNQ1, as well as another E3 ligase called ITCH. Both of these down-regulate Q1 currents as well as surface expression and flow cytometry. They also actually reduce amount of protein, and then we see that primarily they do this by increasing ubiquitin on the channel. They primarily increase K63 chains and a small effect on K48 and the others as well. Distinct lysine residues on the channel were modified by NET42 versus itch, suggesting that even amongst E3 ligases that downregulate the channel, they might be actually different mechanisms by which they may do it. Now, to resolve changes in ubiquitin responses, we turn to our initial assay. We now, in this background, where we have channel along with NET42, the E3 ligase, when we add our linkage specific NDAB, here we now see a ubiquitin responsiveness that's quite similar to what we saw in guinea myocytes, where we actually see a striking increase with K63 as well as K11 in pink, and then it's also rescued by removing all chains, but really no response for the orange one, K2933, or the K48 ones. The functional data nicely correlated with that. Now, with each E3 ligase, we saw more or less the same thing here, except now even the orange one, which removes K2933, also was pretty effective at bringing back surface expression downregulated by itch. So this suggests that there is signaling diversity amongst the E3 ligases in terms of polyubiquitin chains as well. So to note, the highlights of this study is ubiquitin linkages clearly regulate Q1 trafficking dynamics and function. Ubiquitin code changes in response to intrinsic factors like disease-causing mutations, as well as extrinsic factors, cellular context, the type of E3 ligase is present, and so on. Oh, okay. So, switching now to some of the last parts, the ongoing and future projects, I'm now focusing on cardiac calcium regulation in health and disease. So, the insights we gained into polyubiquitin regulation of Q1 channels led us to consider how generalizable is this regulatory code, particularly across different ion channels. So, we've begun to extend this work to the quintessential cardiac L type calcium channel. So, calcium influx through L type calcium channels is a multifunctional signal that controls action potential durations, EC coupling, and gene expression. Dysregulation of channel localization and activity are linked to life threatening conditions like heart failure, arrhythmias, and hypertrophy. So we've gone ahead and adapted our linkage specific end-ups specifically to target calcium channels by fusing a nanobody that directly binds the beta subunit of calcium channels that has previously been published from Henry's group. So we link this nanobody to our linkage specific end-ups and see how if there are any differences in how this channel is modulated and what polyubiquitin signatures underlie it. So we adapted the similar flow cytometry assay where we have still the YFP on the channel so we can measure total expression. Here we saw surprisingly, in sharp contrast to Q1 results, there was really no significant difference in calcium channel expression with any of the end-ups in flow cytometry, at least on YFP levels. Now, we also looked at surface levels by labeling the extracellular Bunker toxin site with 647. So here we saw, surprisingly again, we saw a dominant role actually for removing K63 chains in turquoise, as well as the non-specific one in increasing surface expression of these channels. And looking into the underlying mechanism, both of these seem to operate by slowing down rate of endocytosis of this channel, with really small effects for the other end up linkage specific end ups. Now, looking at function, the non-selective end-up U21 enhanced current output of CAB 1.2 channels in hexyl reconstitutions, as well as in guinea pig cardiomyocytes infected with adenovirus to express the end-up of interest by culturing them. And we see that the relative to control cells, which were infected with the F3, the CAB beta binding nanobody by itself, we saw a shift that suggests at least double of the current. Now, so we are currently investigating if this can be actually used to boost cardiac contractility, especially in the context of heart failure. So, to summarize the whole story, so we saw that distinct post-translational modifications like phosphorylation and ubiquitination can all regulate cardiac ion channel trafficking. And crucial pieces of regulatory information is site of recruitment of the nanobody effector, as well as accessibility to modifiable channel residues, both contribute to how the channel behaves to the and how its trafficking is impacted. The ubiquitin code of ion channels seems to be clearly diverse, alters amongst distinct channel types and cellular contexts, and together this work provides insights on how we can tune these processes to develop therapeutic strategies for distinct arrhythmias and goes beyond the channels we investigated, since the modular nature of the tools can be really adapted to study any channel of interest. So, with that, I would like to extend my sincere thanks to my advisor, Dr. Henrik Kolkraft, for his mentorship and support throughout this project and beyond, to the Dream team for collaborating and for their support through the really hard experiments, my lab members, funding bodies, and thank you so much to SGP for this wonderful opportunity. I'll be happy to take any questions.
Well done, Shri. Lovely data. That
Thank
was
you
a
so
lovely talk.
much.
If we have any questions, I can encourage you to send them in the Q&A. I don't see the Q&A actually. Do we have? Yeah, we do. Okay. Maybe I can start while we get going. So you nicely show, right, that your different dubs can rescue plasma membrane expression of some of the Q1 channels, and that the specific dub might correlate to the specific subcellular compartment in which that mutation gets stuck, right? So I was wondering, when you rescue the channels, can you look at the biophysics of the channel at the membrane and see, is there any correlation between the functional effect of that mutation and where it gets trapped? Does that make sense? So can you functionally classify them into different mutation effects that might govern where they're getting trapped and therefore where they're susceptible to deubiquitination?
It's actually a really thought-provoking question. So, in this study, we didn't really look at function since we were more so interested in the localization. But from previous work from the lab, it's pretty um, the running hypothesis, at least, is the cell seems to be agnostic to functional changes versus interactivity changes. So, a mutation that alters how the channel behaves, opens, closes responses, response to voltage in this case, or uh, those have a functional defect for sure, whether they get to the membrane or not. And then there are another type of mutation where it kind of affects how the channel interacts with its neighbors, how it gets processed for trafficking. And some of them were, when we rescue them with this sort of a tool, especially an endocytosis one, when we were able to prevent rapid endocytosis in a previous paper, it was shown that the function is also rescued. So it seems that in some other cases, of course, you rescue function, but something else may also be wrong with the way the channel behaves. So they seem to be sort of agnostic, which actually provides a nice window, I think, for therapeutic rescue. Depending on the mutation, it may just be a trafficking defect, in which case the tools are great. They can probably help with that. And then sometimes the channel also behaves funky. So in that case, rescuing function does nothing. You
Mhm.
still need the gating modulators, the exciting drugs that are being developed in that arena to be able to make that still work. But it certainly provides a relief for heterologous, like so heterozygous mutations, where you have one copy that doesn't work well, but you have another that works fine. By using the NDubs, we can really extend the shelf life of the functional copy and provide some therapeutic relief that way. So I think those are the angles, but they're kind of complicated and it seems very disease mutation specific. Yeah.
Great. We've got a question here from Pablo Gates. Apologies if I'm getting the surname wrong. Nice work, Sri. I wonder if, in addition to changes in trafficking, there is any change in plasma membrane distribution, increasing clustering or lipid rafts that could indicate that the compartmentalization is altered.
That's a fantastic question and one that we did not look into for the Q1 is very much in my arena to do with the calcium channel. With the Q1, we did even with just the naked eye, we could see that there is some sort of clustering for sure. We just don't know what those are. We have to do more experiments to tell, especially in a cardiomyocyte where you have all the membrane is really not a uniformly distributed area. There's all these niche pockets, so it'll be really cool to see which ubiquitin type codes for maybe a particular domain, which would be really neat. So yeah, things to come in future, I hope.
Question from Dave. Your colleagues, as you alluded, in addition to cardiac diseases, many channelopathies may be addressed via the NDubs you're developing. How long do NDubs last? And presume, ideally, you'd titrate the on and off expression according to disease pathophysiology.
That's a really good question because we don't. So unlike the CRISPR methodologies where you just change it and then you're good for lifetime, with the proteins, of course, it needs to be a continuous thing. And an AAV expression system, for example, for something like this, would have that limitation in terms of expression. We don't know. So we've started to do some of these AAV expressions in mouse, and they seem to last for at least a couple of months. But we haven't really tested that. But really, the long term vision for something like this is to develop small molecules similar to the Protax or the Fix that I showed, where we have small molecule that can recruit an endogenous E3 ligase or D-ubiquitinase, and then another small molecule that preferably binds your channel. would be a drug route to mimic this mechanistic. So that would be mechanism inspired small molecule interventions, which I think might be better for long term.
I was also wondering in your cardiomyocytes, when you modulate the expression of the calcium channel, do you get reciprocal changes in any other channel expression? Is there a logic to how ubiquitination affects this network
Affects
of
the
channels
other.
to match
Um,
depolarization,
we
repolarization?
Absolutely. We actually are really beginning to scratch the surface there. We looked into R by R and we saw that these nicely co-localized still, and there's no aberrant changes, and the action potential durations were still in range, presumably because we didn't see as big an increase in guinea pig myocytes as we did in hex cells. So it's still within physiological range, similar to maybe PK upregulation of channels that I think the cells are able to tolerate it. But we really need to investigate more about other channels to see if those are also upregulated to compensate or not. But yeah.
Layla, do you want to ask your question? You're
Sorry,
muted.
there you
There
go.
you go.
Sorry, really nice talk, Shri. Sorry about
Thank you
that.
so
I'm
much.
having technological issues this morning.
As we
I
all.
was wondering, I was wondering if there's any, if there are any studies about whether the length of the ubiquitin chain that's added to these different residues has any bearing for the eventual outcome of the signal, of the trafficking signal.
It's actually a neat question. The length of the chain.
Or the structure.
I think the structure of the chains for sure. It's sort of so the post-translational modification with ubiquitin and I guess other types too, essentially they alter the surface of the channel itself, right? So if you notice the mutations that I talked about, none of them were really lysine mutations. They just are some other random position, changed the way the channel looked, and then that got recognized and ubiquitinated, presumably, with different polyubiquitin chains. So the shape of the polyubiquitin chains is how many cellular mechanisms are thought to operate to sort of tag a protein for, say, this goes to degradation, this just goes to the plasma membrane now, and so on. And then there's also all the mixed chain types possible. That's like new things about polyubiquitin chain that are coming out. So you can have one really long chain that's all K48 linked, and then you could have suddenly one ubiquitin can also be then ubiquitinated with K63 chain types. So you get really complex ubiquitin chains. Like these are really essentially decorations on the channel that have their own functional interacting partners. But the length itself, this I'm not sure. But the shape, for sure, yes, that's established to be very important. But the nitty-gritties and the details of exactly what shape codes for what is unclear.
Yeah, that's what I was wondering. Okay, thank you.
Yep.
We have another question in the chat from Chandra Prajapati. Since human IKS and guinea pig IKS currents are different in terms of biophysics, do you think that the results you showed is translated to human cardiomyocytes?
That's really a good question because of how cellular contexts really widely vary ubiquitin regulation. The end goal for all of these tools to be able to do them in human-like systems for sure. The Guinea action potential is a bit slower than mouse, making it a reasonable model to test the hypothesis. But when it comes to polyubiquitin regulation, it's really a blank space. What chains are important in a human system is to be determined for sure. Thank you for the question, though. Yeah.
Are there any long QT syndrome variants that introduce a lysine that might introduce ubiquitination?
Mutations that introduce a lysine that introduce ubiquitination. Oh. I have not come across that one. So we have tested some that introduce a lysine, but we didn't really see it have an effect with our end ups. And then there are also mutations where a lysine was mutated out to be an arginine,
Mm.
and that's somehow super stable, but also it's already super stable. And then we thought, oh, that must be because some ubiquitin chain fell off of it or something like that. We threw a bunch of end ups. No, they still increased even more. The sky is the limit, as it seems, for some of them. So, really
That's a shame.
complex network. And there are also other lysine modifiers beyond ubiquitin. So, they could also be sumoylated or netylated.
Brilliant. Lots to unpack. If we
Yeah.
don't have any more questions, I think we'll thank Sri for the great presentation. Thank you everyone for joining, and hopefully we'll see you again on the 5th of May when we have the next seminar from Laurie Alice Peverini, who's a postdoc in the Stefan Pless lab in Copenhagen. So thank you everyone for joining, and enjoy your month. We'll see you next month.
Thank you so much, Connor.
Thanks,
Thank you,
Ree.
everyone.